Hello students, today's lecture is on complement systems and their biological function, alternative pathway. First, we'll have a brief introduction. The complement system is a collection of circulating and cell membrane proteins that play important roles in host defense against microbes and in antibody-mediated tissue injury. The term complement refers to the ability of these proteins to assist or complement the activity of antibodies in destroying lysing cells, including microbes. The complement system may be activated by microbes in the absence of antibody as part of the innate immune response to infection, and by antibodies attached to microbes as part of adaptive immunity. Coming to the biological functions of the complement system. The biological activities of this system affect both innate and acquired immunity and reach far beyond the original observations of antibody-mediated lysis of bacteria and red blood cells. Structural comparisons of the proteins involved in complement pathways place the origin of this system in primitive organisms possessing the most rudimentary innate immune systems. By contrast, the realization that interaction of cellular receptors with complement proteins controls B-cell activities gives the system a role in the highly developed acquired immune system. Thus, we have a system that straddles innate and acquired immunity, contributing to each in a variety of ways. After initial activation, the various complement components interact in a highly regulated cascade to carry out a number of basic functions, including lysis of cells, bacteria and viruses, opsonization which promotes phagocytosis of particulate antigens, binding to specific complement receptors on cells of the immune system, triggering specific cell functions, inflammation and secretion of immunoregulatory molecules. Immune clearance, which removes immune complexes from the circulation and deposits them in the spleen and liver. Now let us have a look on the complement components. The proteins and glycoproteins that compose the complement system are synthesized mainly by liver hepatocytes, although significant amounts are also produced by blood monocytes, tissue macrophages, and epithelial cells of the gastrointestinal and genitourinary tracts. These components constitute 5% by weight of the serum globulin fraction. Most circulate in the serum in functionally inactive forms as proenzymes or zymozymes, which are inactive until proteolytic cleavage, which removes an inhibitory fragment and exposes the active site. The complement reaction sequence starts with an enzyme cascade. Complement components are designated by numerals C1 to C9, by letter symbols example factor D, or by trivial names example homologous restriction factor. Peptide fragments formed by activation of a component are denoted by small letters. In most cases, the smaller fragment resulting from cleavage of a component is designated A and the larger fragment designated B. For example, C3A, C3B, note that C2 is an exception. C2A is the larger cleavage fragment. The larger fragments bind to the target near the site of activation, and the smaller fragments diffuse from the site and initiate localized inflammatory responses by binding to specific receptors. The complement fragments interact with one another to form functional complexes. Those complexes that have enzymatic activity are designated by a bar over the number of the symbol. Example, C4B2A, C3BBB. Now let us see the process of complement activation. This figure outlines the pathways of the complement activation. There are three major pathways of complement activation. The alternative and lectin pathways are initiated by microbes in the absence of antibody, and the classical pathway is initiated by certain isotypes of antibodies attached to antigens. The early steps culminating in formation of C5b can occur by all the three pathways. The final steps that lead to a membrane attack are the same in all the pathways. Now let us see the alternative pathway. The alternative pathway generates bound C5b, the same product that the classical pathway generates. But it does so without the need for antigen-antibody complexes for initiation. Because no antibody is required, the alternative pathway is a component of the innate immune system. This major pathway of complement activation involves 4 serum proteins: C3, factor B, factor D, and properdin. The alternative pathway is initiated in most cases by cell surface constituents that are foreign to the host. For example, both gram-negative and gram-positive bacteria have cell wall constituents that can activate the alternative pathway. The intermediates in the alternative pathway for generating C5b are shown schematically in the diagram. In the classical pathway, C3 is rapidly cleaved to C3a and C3b by the enzymatic activity of C3 converters. In the alternative pathway, serum C3, which contains an unstable thioester bond, is subjected to slow spontaneous hydrolysis to yield C3a and C3b. The C3b component can bind to foreign surface antigens such as those on bacterial cells or viral particles, or even to the host's own cells. The membranes of most mammalian cells have high levels of sialic acid, which contributes to the rapid inactivation of bound C3b molecules on host cells. Consequently, this binding rarely leads to further reactions on the host cell membrane. Because many foreign antigenic surfaces, example bacterial cell walls, yeast cell walls, and certain viral envelopes have only low levels of sialic acid, C3b bound to these surfaces remains active for a longer time. The C3b present on the surface of the foreign cells can bind another serum protein called factor B to form a complex stabilized by magnesium ions. Binding to C3b exposes a site on factor B that serves as a substrate for an enzymatically active serum protein called factor D. Factor D cleaves the C3b bound factor B, releasing a fragment BA that diffuses away and generating C3BBB. The C3BBB complex has C3 convertase activity and thus is analogous to the C4B2A complex in the classical pathway. The C3 convertase activity of the C3BBB has a half-life of only 5 minutes unless the serum protein properdin binds to it, stabilizing it and extending the half-life of this convertase activity to 30 minutes. The C3BBB generated in the alternative pathway can activate unhydrolyzed C3 to generate more C3b autocatalytically. As a result, the initial steps are repeated and amplified so that more than 2 × 10⁶ molecules of C3BB can be deposited on an antigenic surface in less than 5 minutes. The C3 convertase activity of C3BBB generates the C3BBB3 B complex, which exhibits C5 convertase activity analogous to C4B2A3B complex in the classical pathway. The non-enzymatic C3b component binds C5 and the BB component subsequently hydrolyzes the bound C5 to generate C5a and C5b. The latter binds to the antigenic surface. The three complement pathways converge at the membrane attack complex. The terminal sequence of complement activation involves C5b, C6, C7, C8 and C9, which interact sequentially to form a macromolecular structure called the membrane attack complex (MSC). This complex forms a large channel through the membrane of the target cell, enabling ions and small molecules to diffuse freely across the membrane. The end result of activating the classical, alternative or lectin pathway is production of an active C5 convertase. This enzyme cleaves C5, which contains two protein chains, alpha and beta. After binding of the C5 to the non-enzymatic C3b component of the convertase, the amino terminus of the alpha chain is cleaved. This generates the small C5a fragment which diffuses away and the large C5b fragment which binds the surface of the target cell and provides a binding site for the subsequent components of the membrane attack complex. The C5b component is extremely labile and becomes inactive within 2 minutes unless C6 binds to it and stabilizes its activity. Up to this point, all the complement reactions take place on the hydrophilic surface of the membranes or on immune complexes in the fluid phase. As C5b6 binds to the C7, the resulting complex undergoes a hydrophilic amphiphilic structural transition that exposes hydrophobic regions which serve as binding sites for membrane phospholipids. If the reaction occurs on a target cell membrane, the hydrophobic binding sites enable the C5b67 complex to insert into the phospholipid bilayer. If, however, the reaction occurs on an immune complex or other non-cellular activating surface, then the hydrophobic binding sites cannot anchor the complex and it is released. Released C5b67 complexes can insert into the membrane of nearby cells and mediate innocent bystander lysis. Regulator proteins normally prevent this from occurring, but in certain diseases, cell and tissue damage may result from innocent bystander lysis. Binding of C8 to membrane-bound C5b67 induces a conformational change in C8 so that it too undergoes a hydrophilic amphiphilic structural transition, exposing a hydrophobic region which interacts with the plasma membrane. The C5b678 complex creates a small pore, 10 angstrom in diameter. Formation of this pore can lead to lysis of red blood cells but not of nucleated cells. The final step in the formation of the MSC is the binding and polymerization of C9, a perforin-like molecule, to the C5b678 complex. As many as 10 to 17 molecules of C9 can be bound and polymerized by a single C5b678 complex. During polymerization, the C9 molecules undergo a hydrophilic amphiphilic transition so that they too can insert into the membrane. The completed MSC, which has a tubular form and functional pore size of 70 to 100 angstrom, consists of C5b678 complex surrounded by a poly C9 complex. Since ions and small molecules can diffuse freely through the central channel of the MSC, the cell cannot maintain its osmotic stability and is killed by the influx of water and loss of electrolytes. Now coming to the biological consequences of complement activation. Complement serves as an important mediator of the humoral response by amplifying the response and converting it into an effective defense mechanism to destroy invading microorganisms. The MSC mediates cell lysis, while other complement components or split products participate in the inflammatory response, opsonization of antigen, viral neutralization, and clearance of immune complexes. Many of the biological activities of the complement system depend on the binding of the complement fragments to complement receptors, which are expressed by various cells. In addition, some complement receptors play an important role in regulating complement activity by binding biologically active complement components and degrading them into inactive products. The membrane attack complex can lyse a broad spectrum of cells. The membrane attack complex formed by complement activation can lyse gram-negative bacteria, parasites, viruses, erythrocytes, and nucleated cells. Cleavage products of complement components mediate inflammation. The smaller fragments resulting from complement cleavage C3a, C4a, and C5a called anaphylatoxins bind to the receptors on mast cells and blood basophils and induce degranulation with release of histamine and other pharmacologically active mediators. The anaphylatoxins also induce smooth muscle contraction and increased vascular permeability. C3b and C4b binding facilities opsonization. C3b is the major opsonin of the complement system, although C4b and C3b also have opsonizing activity. The amplification that occurs with C3 activation results in a coating of C3b on immune complexes and particulate antigens. The complement system neutralizes viral infectivity. Large viral aggregates reduce the net number of infectious viral particles. Antibody bind to viral particle creates a thick protein coating that neutralizes viral infectivity by blocking attachment to susceptible host cells. The complement system clears immune complexes from circulation. Erythrocytes play an important role in binding C3b-coated immune complexes and carrying these complexes to the liver and spleen. In these organs, immune complexes are stripped from the red blood cells and are phagocytosed, thereby preventing their deposition in tissues. Conclusion. The complement system is a collection of circulating and membrane-associated proteins that are important in defense against microbes. Many complement proteins are proteolytic enzymes, and complement activation involves the sequential activation of these enzymes, leading to the generation of effector molecules that participate in eliminating microbes in different ways. The complement cascade may be activated by any of the three pathways. Activated complement proteins become covalently attached to the cell surfaces where the activation occurs, ensuring that complement effector functions are limited to the correct sites. The alternative pathway is triggered when some complement proteins are activated on microbial surfaces and cannot be controlled because complement regulatory proteins are not present on microbes but are present on host cells. The alternative pathway is a component of innate immunity. Thank you.